Outcomes Public Health Economics Research Center, funded by India Alliance, which is India Alliance is by Department of Biotechnology, Government of India, and Wellcome Trust of UK. Today we have a very important speaker, Isidore Rigoutsos from Jefferson University. Today's session will be moderated by Dr. Anthony Vipin Das from LV Prasad Eye Institute. I'll briefly uh, uh, just introduce Vipin before he takes over the proceedings. So Vipin is a consultant ophthalmologist at LVPI, and he heads the iSmart EMR, which is a well-renowned electronic medical record system, not just in India, but across the world. And it has clocked over 7.5 million patient consults. And he also collaborates with Jefferson on a lot of big data studies and analytics in healthcare. So over to you, Vipin, for the proceedings.
Thank you, Dr. Raja. And it gives me great pleasure to introduce a mentor and collaborator to me. Dr. Isidore Rigoutsos is the founding director of the Computational Medicine Center at Thomas Jefferson University. He joined Jefferson in early 2010 as a professor with an appointment in the Department of Pathology, Anatomy, and Cell Biology. He also has joint appointments in the Department of Cancer Biology and the Department of Biochemistry and Molecular Biology, and is a member of the university's Sidney Kimmel Cancer Center. Prior to joining Jefferson, Dr. Rigoutsos worked at IBM's Thomas J. Watson Research Center for nearly 18 years. There, he co-founded the Computational Biology Center in 1992. He also founded the Bioinformatics and Patent Discovery Group in 1998 and managed it until his departure from the company. In parallel to his IBM tenure, Dr. Rigoutsos was for a decade a visiting lecturer at MIT's Department of Chemical Engineering, where he taught graduate level classes and summer professional courses in bioinformatics and co-supervised PhD students. Dr. Rigoutsos' involvement in the field of computational biology spans more than 25 years. Since 1996, his efforts have revolved around the theoretical and practical aspects of pattern discovery and the design of pattern-based solutions to a variety of problems from genomics, genetics, molecular biology, and medicine. It gives me great pleasure to invite Dr. Isidore Rigoutsos to deliver his talk this evening. Dr. Rigoutsos?
Thank you, Vipin. Thank you, Raja. Thank you, everyone, for the kind words and the invitation to present. I understand, we are doing all of this under dire circumstances. Everyone is looking forward to better days for India and for everyone, really. I'll try to be mindful of the time limit. I understand there is a curfew we need to worry about. Let's see how things go. Maybe it's best if we leave the questions for the end. Can you see this?
Yep.
All right, very good. So I will move to a very brief description of who we are. As you can see, we are young and old and in between. We are training the babies into bioinformatics. We start them early. What you see here is basically the Computational Medicine Center members. It's a picture just before the pandemic. In addition to thanking them for generating much of the work that I'll be presenting, I want to thank a whole number of other people that over the years helped the efforts. And as Vipin mentioned, I've been in this particular subfield for quite a few years. So today I'm hoping to be covering several seemingly diverse topics. I enumerate them here and I'll embark on each one in turn. So let us begin with the first one, which is something that people usually confuse. They confuse in particular searching with discovery. And these are two very different objectives, and computationally they are also very, very different. And of course, there is the concept of clustering where you put like things together, and I'll discuss how this comes into play as well. So what, let me start with the question. When we search for something, what exactly do we do? What do we look for when we search? And what I'm trying to say here is when you search for something, you have a reference. You're holding an object, a word, something, and you try to look for additional instances of that in a database. So the item you are looking for is given. There is a definition, there is a description, you know what it looks like. It look like my object. You double the size of the database, you take twice as long. This is not the case though when you mine. When you mine, what do you look for? Just before we began, there was discussion of mining. What does the goal look like? And the answer is, we don't know. We just know there is merit in doing it. There is a database, there is something hidden in it, but what are we looking for exactly? The description of it is not given. And clearly, intuitively, you can think, you can see why it would be much more difficult to mine. You may or may not be familiar with gene expression arrays. These were all the hype in the 90s and early 2000s. And by that, I mean, it was the technology of choice. You had an actual physical device that could ask 40,000 questions. And each question was, what's the expression of the gene X? And there were 40,000 genes on it, coding and non-coding genes. And what you see here is a clustered array. These are readings. There is about 6,000 rows. I'm not showing all 6,000. And there is about 80 columns. Every column is a time point, and what you see in a given row is how the gene's expression changes as time goes on. Green is low expression, red is high expression. What you notice though is adjacent rows look like one another, and there are groups of different rows. So why is clustering not enough for the purposes of mining? In the early days, people did use clustering for mining, but it's not enough. Why? For the simple reason that there may be some rows that share a short region and nothing before, nothing after. If you're looking for things that look globally the same, you're going to miss this cyan region that I'm showing here. And I'll return to an example of that in a little bit. So pattern discovery, I will introduce you to two basic versions of pattern discovery. And the first one is you're given records of variable length. Think of words in a natural language, and they're chained into phrases. So every row is a phrase. You have phrases with different numbers of words, and now you're looking for something. What does it look like? Again, you do not know, but what you do know is it repeats. And it can be short, it can be long. So the obvious questions is, how many colored blocks can you find in a given piece of text I give you? Where do they begin? Where do they end? How many copies do they have? So these are the obvious questions, and ahead of time, you do not know the definition of the red, the yellow, and the cyan blue, or if there is more than 3. The second version is the kind of version that we encounter when we mine a database like eyeSmart. You have many, many rows. These would be your patient visits. You have columns. These would be the things you ask of a patient during a visit. You can have millions of rows. You heard moments ago that iSmart is over 7 million records. You can have many columns. There can be hundreds of columns depending on how granular you want to be. What do you look for here? What you look for is subsets of patients that agree on some of their answers to the questions that you care about as a clinician. The problem, though, is you do not know who these patients are, and you do not know what attributes they agree on. So essentially, when you do mining, you have to pose the question and answer it too. And ahead of time, you don't know what the question is. So no matter whether it's version 1 or version 2, what you rely on is counting. You simply count, and the goal is to count as quickly as you can to make it practical. So let's go back to this. I will give you an example. This is a stream of Greek letters. For all you know, it's nonsensical. It's a nonsensical stream. Ahead of time, you have no sense of whether there is value here, whether there is something to be mined. Intentionally, I removed any spaces that might have been there, so you do not have a sense of where things begin and end. The obvious questions are, is it random? Maybe, maybe not. Not knowing the language, you cannot answer the first question. If it's not random, then what's hidden in it? And if it's hidden, is it short, long, and of course, the number of copies. I will show you some of the things that repeat. And you can, this is something you can do with data mining, and I'll show you how you do that. But here's the problem. The problem is you find these combinations of letters, but what do they mean? I give you some meanings here, but if I didn't, you wouldn't know what the combination of letters on the left means. The first one means the God. O is an article in Greek. And you can see there are more involved, there are more involved combinations of letters. And in the general case, I give you a combination, you don't know what it means. But here's something interesting. There is a very interesting game you can play in addition to what I'm showing you here. If you pay attention to the Greek strings that I give you, you'll see that some repeat. So these repeat. So if you have figured out the meaning for, let's say, the fourth combination of letters from the top, and you have the meaning, and you realize that the fifth one agrees on something, then you know that the black string of letters must mean thusly and. In other words, once you figure out something, you remove it, you remove the corresponding meaning. What's left from the meaning must agree with what's left with the string of letters. So you can play this game and start adding sense to many things you find. And in case you're wondering, the text is from the first chapter of the book of Genesis in the Ancient Testament. So let me give you briefly a description of an algorithm that we published many years ago, more than 20 years ago, and it proved to be a terrific cookie cutter, a terrific tool for us, because over the 20 years, we've used it in numerous settings, in particular in biology and medicine. So the algorithm made a very simple observation, which in hindsight is very intuitive. If there is a long phrase in a piece of text I gave you that appears k times, then short short combinations of letters from that phrase must appear at least k times. It's a very straightforward observation. And the second sentence, the fact that the short things can appear at least k times, is the key to the algorithm, and I'll show you how. So the algorithm has two phases. In the first, you collect all short combinations of things that repeat. And that's a very easy and quick thing to do. The second, you put them together into increasingly longer phrases. This is all done automatically, of course. So let's go back to a stream of English letters. There is something hidden here, and we'll find it. So we're going to look for patterns that appear at least 3 times and contain at least 3 consecutive letters. So I'm going to show you the patterns. This stream of letters has over 1,000 letters, and for every triplet, I'll tell you how many letters from the beginning it appears at. So here's the first triplet of letters. You'll find it at position 193, 635, and 1112. Now I'm going to show you the rest of the triplets that appear 3 or more times. And these are actually counted from this particular text. There is nothing else that appears 3 or more times. So how do you put them together? If you pay attention, you'll see that some triplets end in 2 letters, and the same 2 letters are at the beginning of another triplet. So this is the key observation. So let's take 2 of these and put them aside. You can see the common 2 letters being PP. Now, if you notice the left one, APP begins at position 19, but there is one instance of PPR beginning at position 20. Aha, now you can combine them. You can make something longer. So you say, since these are congruent, I can combine them with APPR, and I know APPR begins at position 19. And you can continue playing this game, and again, and again. Now, notice what happened. We have APPR, which is 1 longer than what we started, and we used up all of the PPR instances. So we do some cleanup, and this is basically what we have at this point. But if you notice, what we have is similar to what we had before we started the convolution. So you keep on playing this game until you run out of the ability to make something longer, or what's hanging from a given combination of letters has fewer than 3 things. And once you complete this game, you'll find that there are 4 copies of the word approach, 4 of the word poach, 3 of the word ache, 3 of apple, and of course you have suffixes that you expect. Now let me skip to this now. So I mentioned that here what we're looking for is something along these lines. So many years ago, actually before I give you the example, I want you to think about the problem intuitively. If, for example, we had only 15 rows and 1,000 columns, and we tried to look for subset of rows that agree on some columns. Ahead of time, you don't know what the columns are, but all of the groupings can come from only 15 rows. And if you do the combinatorics, there is only 2 to the power of 15 combinations of rows, which is not a big number. It's only 32,000 and change. So you enumerate the subsets, and for every subset, you check which columns the records agree on. Now, it's not obvious, but what I just described, you can do on your laptop in maybe a matter of 10 seconds. It sounds complicated, but 10 seconds are sufficient to do mining on something that has very few rows. Where things get really hairy is when the number of rows is in the thousands, the hundreds of thousands, in the millions, such as the case of eyeSmart. So the combinatorics there becomes prohibitive. Still, we can actually tackle it with the algorithm that I just described. So I mentioned before that clustering is not enough because you cannot find this kind of pattern. Now, in this particular gene expression array, there is such a pattern that we were able to find, and I'll show it to you now. This is the pattern. What you look at is time going from left to right along the x-axis. It's in hours, and the y-axis is the expression of a gene in log 2. So you can basically see that for quite a lot of time points, these genes, every curve that you see here is a gene. They are minding their own business. They increase, they decrease, they stay flat until they get to this time point where they all work together. They decrease together, they increase together, etc. And then after this interval, they mind their own business again. So mining will be able to find this, but not clustering. So clearly early on, we were interested in applying this to a medical setting. So we started many years ago at the time that electronic medical records were not a frequent occurrence. So what you see here is a paper that we published in 2005. By then, we had spent a year and a half to 2 years analyzing the data. So we were embarking on electronic medical record mining about 17 years ago. We were fortunate enough to have met William Naus from the University of Virginia. And he had access to the state's health system records, and he gave us access to almost 700,000 patient data set, patient records. Think of a matrix, 700,000 rows, and for every patient, we had about 120 columns. So we played the game that I described. Sure enough, we found combinations of observables that were expected. They were in the literature, they were known to the medical practitioners. Where it got interesting was we found also things that were not known. And here's an example. So this we wrote in the paper, and we said, looking at all these records, we find very strong evidence for an unlikely combination. This combination doesn't appear by chance. And the combination was anemias plus omeprazole plus hypertension. Omeprazole is the generic substance in Prilosec. This is a substance that inhibits proton pumps in the stomach, and it's used to essentially control acid reflux. We looked through PubMed. At the time, there were no articles that were describing any of these combinations, and we wrote so in the paper. We moved on. And then 6 years later, the Food and Drug Administration of the United States puts out a safety announcement. And you can see from the header that essentially they were warning clinicians that if a patient is on sustained proton pump inhibitors for 2 or 3 years, they're going to develop anemias in this case, low magnesium levels. And then a year later, again, the Food and Drug Administration issued it wasn't a warning because all of the evidence was self-reported by patients, and they said Prilosec has been associated with elevated blood pressure. Essentially, these two warnings explained what we found 7 years earlier. It turns out in 2013, a basic research paper came out that showed that people who are on long exposure to omeprazole will develop high blood pressure, and they showed that this inhibition of the proton pump can actually lead to heart attacks. And they showed the actual mechanism of how this happens. And that was in 2013. So essentially, by mining, we were able to find something that first we couldn't explain, but it made perfect sense years later, once we had additional knowledge. Which of course brings us to more modern medical record systems. And this is work that we have been doing with Vipin and his team. We were given a snapshot of eyeSmart. At the time, it had a little over 2 million records. And in order to remain focused, we focused on pterygium. One of the conditions, there were about 183,000 records of individuals with that condition. And for every record, we had 30 variables that we zoomed on. And we did mining. We found over 600,000 patterns that we were able to combine in 3,150 supergroups. What do I mean by supergroups? Many patterns were beginning with being general and were becoming a little more detailed in smaller groups of patients, but the essential pattern remained the same. So out of the 183,000 records, we distilled 3,000 essential patterns. The patterns involved between 14 patients and 151,000 patients. And they could involve as many as 13 variables of the 30. I will give you an example. Obviously, going through all of these data and making sense of it is a time-consuming endeavor. But essentially, the answers, the gems, if you wish, are in this one Excel that we have built out of this analysis for one condition. And obviously, there are other conditions in eyeSmart that we can extend this to. I'll give you an example. We have, this is a very simplistic example, but it gives you a sense of what we can do. We found that there are 8,875 records that come from individuals who live in a rural setting, are adults, female. They come from a specific district in the state of Telangana and have, the cornea is clear. And these individuals have an odds log2 ratio of 2 compared to chance to develop pterygium. Then for them, there is another group of 10,000 individuals with a different combination. They are male and they are from a different state, but for them, the odds ratio is negative. And you can see how this plays out. Essentially, we figured out subgroups of patients that have different levels of propensity for pterygium, either enhanced or diminished propensity compared to chance. And the mining allowed us to basically figure out what it is they all share. And now I'll move to biological examples so that you can see the diversity of the applications that are possible. So I showed you this before. This is the first, it's the opening of the book of Genesis in Greek. And what we asked ourselves, and again, that was many years ago, we first asked that question 16 years ago. And the question was, what if we play this game with nucleic acid sequences? Messenger RNA that encodes a protein. What if we had many messenger RNAs? Can we figure out groupings of things? So we did precisely that. And let me show you an example of 9 such messenger RNAs. So I have colored different things. I'll zoom in and I'll explain what it is you are seeing. But as you look at it from afar, you can see the same colored block appearing. So you can see this colored block, AGTA, GCT, appearing here in the same sequence twice, appearing in another sequence, and another, and so on and so forth. If you focus on the third sequence, it's preceded by a purple block, but in the first sequence, you don't see that purple block. Essentially, what you're seeing is combinations of blocks of letters that appear frequently, sometimes together, sometimes with different neighbors, essentially analogous to what we do with natural languages. So I said, great, let me zoom in now and show you the detail. Whether you see something colored or you see it in black letters, it's actually a word. It appears frequently. I've only colored some of them because it's very difficult to color them by hand. Now you see numbers such as 53, 6, etc. That means 53 means there are 53 letters that I cannot break into words, and they don't repeat. So these are unique. Essentially, 53 says, I have no idea what's going on here. Before the 53, I have a block. After the 53, I have a block, and so on and so forth. So this is, again, the picture. Great. So we said, fine, we can play this game, and we played it. And we found words, and there were many. And we used the Greek word for dense to refer to them. We called them pyknons. So how many did we find? We found 209,000 in the human genome, and we found 120,000 in the mouse genome. And of course, the obvious question is, well, do humans and mice use the same words? And the answer is no, they don't. We basically had only 3,000 pyknons in common. Still, we had the words. Now for the bad news. The bad news was we had no idea what the words mean. We knew they were sensible because if a messenger RNA reuses a certain block of letters, that means it's doing something. But we had no idea what it is it was doing. Years went on, and we were able to show that these words are relevant for different things. We linked them to metabolism, we linked them to DNA repair, but for the individual words, we weren't able to actually find a setting where they mattered. Until one of my PhD students, Aris Telonis, got interested in this toward the end of his PhD thesis, and that led into a 2-year endeavor. And here's what he was interested in. He looked at the very early stages of an embryo, from the fertilized egg, the zygote, to the early blastocyst. So we were focusing on these 2 time points that are separated by about 1 week. We focused specifically on genes that are high in the zygote and decrease in abundance toward the blastocyst, and the opposite, genes that are not present in the zygote but increase in the blastocyst. We found them. We did this for human and mouse, and we found that the genes that are high in the zygote are long and encode signaling proteins. And the ones that are high in the blastocyst are short and encode a different set of proteins, mostly ribosomal proteins and metabolic enzymes. The revelation for us was something else. We asked, are there any pycnons that we can find in these data sets? And turns out that the genes that are active in the very first hours and very first few days of a fertilized egg are jam-packed in pycnons at a statistically significant level. The p-values were 10 to the minus 5. And as you can see, these pycnons are about a third of everything we had accounted for. Essentially, a third of these words seem to be active in the first few days of a fertilized egg. But once you get to the blastocyst, they seem to be gone. So now we know where to look. We didn't anticipate something like this, obviously, but because most of the work we are doing comes from cancer. It's at a later point in time, different types of cells, and this link to the pycnons is not very prevalent in cancer, but in differentiating cells, it is. And finally, I want to spend a few minutes discussing about data mining in general and precision medicine. This is a new line of work in the sense that we didn't start 15 years ago. It goes back about 8 years, and it was spurred by an accidental discovery. So I will introduce you to a set of special RNAs called microRNAs. If you don't know anything about them, that's okay. I will introduce them briefly. So what is a microRNA? A microRNA is a piece of DNA that produces a very specific molecule. So this molecule, and you can see an example of this, the parental DNA is about 100 letters long. So this molecule has a specific peculiarity. Sorry. Once it's made into RNA, it folds into a shape that looks like a hairpin, which you can see here. It's basically this shape again and again and again, with a few bulges here and there. So there is a process that people discovered about 30 years ago that will take this shape and produce a short product, this purple molecule, which is very short. And we refer to this molecule as, we refer to this molecule as the microRNA, the mature microRNA. And why do we care about studying these very short molecules that are about 20 letters long? We care about them because they become part of a regulatory complex that controls protein abundance. In all cells, all time points of a person's life, in health and in disease. The very first paper that linked them to disease showed that if you remove one of these sequences, the 100-letter long mother, you develop leukemia. And they showed this in mice. Now, what is the problem with these sequences? The problem is our understanding was incomplete. New technology has shown that these molecules don't make one product, which I show here highlighted in gold. They make multiple. And you can see them aligned to this one product we have been studying for 20 years. people's initial reaction was, oh, you're probably looking at a technological problem, a technological limitation that's basically trimming these molecules at either end, or you're seeing at the enzyme misfiring and not cutting the short molecule properly. But, you know, just focus on the one that's highlighted in gold and forget about the others and move on. Well, it turns out that these molecules, these other molecules from the same locus, are not degradation products. In fact, we were able to show that their production is regimented. What do I mean by that? You take samples from different people from the same tissue, and you always see the same combination of daughters. But perhaps most importantly, for the purpose of personalized medicine, was that we were able to show, and my group was first to show this, we were able to show that how many of these variants you make at a given time and how much you make of a given variant depends on your biological sex, depends on population of origin, race, ethnicity, as well as tissue type, tissue state, and disease. In other words, you have to know all of these variables before you can study any of these molecules in a given setting. And the reason this matters is we were able to show experimentally that different variants from the same mother, the same parental precursor, will target different protein coding genes. In other words, each one has a mind of its own and acts independently, leading essentially to a push and pull, a tug of war where at the center is your protein producing messenger RNA, and different daughters from different microRNAs are pulling it in the direction they please. And whether one or other wins depends on your biological sex and your ancestry. And we showed recently depends on age as well. And I'll show you something complementary very quickly. It's something you'll see a pattern repeating here. So I'm sure you are all familiar with transfer RNAs. We've known of them for over 65 years. And the idea is my DNA will make a transfer RNA, and that transfer RNA is used to produce proteins. I highlighted here the anticodon and the amino acid that is given to the nascent protein that's produced by the ribosome. So the picture for many years was I have one piece of DNA, it makes one molecule, a tRNA. And again, we were wrong, we the community, in the sense that this knowledge was incomplete. We know now that what happens is you do get your one tRNA, but you also get a lot of different fragments, and these fragments belong to specific categories. And I color-coded the categories here. My group was able to discover and report for the first time this category at the bottom, the ITRFs. And of course, you know, the question is, are you looking at degradation products? And the answer is no. We showed again exactly the same kinds of properties that we showed for microRNAs. We published this for the first time. We published this for the first time about 6 years ago. And you can see again the same dependence. How you cut a tRNA into fragments again depends on who you are. It depends on your biological sex, your ancestry, and it also depends on the tissue. And the reason you care about these molecules is because just like microRNAs, they regulate the abundance of proteins. So essentially, you're looking at regulators again. that no one in the community suspected for 60 years. And now we know they are there. Not only that, but they are affected by who we are. And of course, the question is, how many molecules are we talking about? And the answer is many. For microRNA isoforms, we have published more than 10,000. For tRNA fragments, we have published more than 26,000. And there is a third category that I didn't discuss here that derive from ribosomal RNAs and have exactly the same properties, meaning they depend on biological sex, and they depend on ancestry and tissue and disease subtype. And they are also regulatory in nature. They regulate protein abundance. And for them, we have published more than 18,000. So in keeping with what we've been discussing, the situation is essentially, here is more than 50,000 words whose meaning we do not know yet. Clearly, it's going to be a lot of work to understand it, but we can use them in the meantime for diagnostics, for therapeutics that are specific to individual people. And with that, I just want to summarize everything. Essentially, I hope I convinced you that data-driven science is effective. In my opinion, the second point is perhaps the most important one. It is powerful. It's unconstrained in the sense that it will find things that are not in the books. And more importantly, it's unbiased. In other words, it doesn't matter what I believe the result ought to be. If the data says differently, that's it. I have to follow the data. And for the two categories I mentioned, the isomers and TRFs, they have terrific potential to become high quality biomarkers. We have a series of papers that show this and also show the potential as therapeutics. So in a nutshell, we, the community at large, have been talking about precision medicine. And now we are beginning to generate tools and interaction points, contact points, if you wish, that will allow us to make this a reality. And with that, I want to thank all of you for taking the time to listen to my presentation, and Vipin and Rajaf and LV Prasad for the invitation. Thank you.
Thank you so much, Dr. Rigoutsos, for a wonderful lecture and a very informative and an eye-opening lecture. We do have a few questions coming in, so I'll request our attendees to kindly put in your questions in the chat box, and we will be able to answer those questions. So to begin with, we have Dr. Raja with a few initial questions. His first question is, what kind of data structure is required for data mining and pattern discovery?
Excellent question. Speaking from experience, it's very difficult to spell out a recipe. What's interesting here is this is where we spend most of our time. And I'll give an example with iSmart. So iSmart, you can give us a set of records that look like a matrix. The problem is, because the records are contributed by different attending doctors, different clinicians, the words are not necessarily normalized. One could use a different word to say the same thing, or they can use a phrase. And there is an initial phase, and Vipin's team and my team went through a lot of pain there to normalize the terms that are being used. Once you normalize it, then things are much more amenable to mining. In the case of nucleic acid sequences, amino acid sequences, things are a little better because the community has imposed a structure and format going back 25 years. So in In biological settings, things are pretty straightforward because there is a structure already that everyone needs to follow if they want to report something. For medical records, it's less structured and more painful. So um, expect to put a lot of time into cleaning up the records in order to make the machine accessible.
Yes. The second question that he has, thank you, Dr. Rigoutsos. The second question that Dr. Raja has is, should the patterns which we get be subjected to additional statistical analysis for confounders?
Yes, of course. The problem, it's a catch-22 here. By virtue of having a pattern, you're basically reporting correlations. So all of a sudden you have variables that you didn't know are correlated, but they are correlated. So you need to go to a group of people who are outside the control people and find many that have the same pattern and count the incidents. You cannot assume that the different variables are independent. You could, but the reality of having found a pattern of correlation says they're not independent, right? So it becomes a little tricky of how do you actually estimate the probabilities. The cleanest way, and something that we did in the case of the work with Vipin's team, is to basically find people who do not have the disease. So if you work with Teiresias, you have another almost 2 million people in our database that don't have it. So you use those as controls.
Thank you, Dr. Rigoutsos. So again, in essence, Dr. Raja's question is, can the patterns ever be misleading? In essence, how do we get to trust the patterns? Can they also be misleading based on the input of information as well?
They can be misleading in the general, the quick question is no, they cannot be misleading. However, having said that, they can be misleading in the sense that they do not provide any valuable knowledge. If you think of my Greek text example, if you play the same game with English, the most frequent pattern you will find is probably the article the, the. By virtue of frequency, it's important. By virtue of meaning, it's not. So a pattern always needs to be coupled to the meaning because it's the two of them that make or don't make sense.
All right, thank you. We have a question from Mr. Murlidhar. Are the concepts and techniques of principal component analysis useful in such studies?
Partially. Essentially, you can think of principal component analysis as clustering. It's a form of clustering. It will look for global similarities across the points that you are considering. It will miss subsets of things like the gene expression array that was all red and all of a sudden you see green and all red. A principal component analysis approach will be sidetracked by the fact that there is so much red and very little green. And basically it will say there is nothing here because globally looking, these genes are different. You see the similarity only locally and for a very small time interval.
Thank you, Dr. Rigoutsos. I do have one question that might be relevant to our group today. The IRIS registry, which is, of course, the world's largest registry on ophthalmic diseases from the United States. From at least Asia, we now have eyeSmart, and of course, a couple of national disease registries in Europe. And I know you are one of the 5 investigators who also has access to IRIS registry to work on it. How has been your experience so far in working with such large national data registries, such as the IRIS in the United States?
I haven't had an experience, to be honest. Neither has my team at the Computational Medicine Center. The way IRIS has been structured is to allow a hypothesis. And then you look at the results, and then you repeat this, and you repeat this. So this is very different than what I described. In our case, we say, this is the entire database. Is there anything that the data supports? When you do the hypothesize and verify in population researchers, essentially do that. They create a model, they assume 10, 12, 15 variables, and they say, well, is my model able to explain? And they go and check the model. They get an answer, they modify the model, repeat. But that's driven by one's knowledge, the contents of books, and it's colored by potential personal biases in how to create a model. On the contrary, what we did, meaning your team, Vipin, and my team with eyeSmart, was totally unconstrained, totally unsupervised. Go in, whatever the data says goes together, report it. You still have to address the question that Raja put forth, which is, well, you need to evaluate the statistical significance, and we have ways of doing that. And at the end of the day, you need to associate meaning. In the case of eyeSmart, that's easy because it's a very well maintained, very well controlled, very well structured database. It makes it trivial. But remember, I think the privilege and the luxury you have with eyeSmart is you started creating it in a very regimented manner from day one. And that, in my opinion, is invaluable. And that's why, when you and I, Vipin, started talking more than 2 years ago, I'm like, oh, yes, that's the way it ought to be. So from day one, you designed it with an eye toward what we were discussing in the last hour. IRIS is an aggregate by different people, different institutions, different points in time. And of course, at that point, you face the other point that Raja brought up, which is how do you need to clean up and prepare the database now, right? Especially when it's contributed by multiple people.
Yeah.
Perhaps the last constraint that I see with IRIS is we are not allowed, even in a de-identified way, to access the entire iris in-house. Because for us to be able to play this game, we actually need to have all of iris and do a super mining, which will take weeks of a high performance computing system. And there is no hypothesize and verify here. But the way it's structured, IRIS is structured currently, it forces a hypothesize and verify paradigm.
Thank you, Dr. Rigoutsos, for your invaluable insights for us. So we are almost running out of time, but there is a final question from one of my colleague, Mr. Ganesh. His question is, what basic software knowledge is required to get started for beginners? Pattern recognition.
For beginners.
101, yeah.
Well, so let's start. Unix would be good. Ability to program in C or any other language that allows for speed would be good. And then a course such as knowledge discovery and data mining. We actually put together a course and we taught for the first time last summer at Jefferson. for our students, because the questions that I discussed with you today are beginning to be posed by more and more of my colleagues, students, and faculty. So we need to educate the people. So essentially, Unix, a programming language like C, and the basics of data mining and knowledge discovery.
Great. So if there are no questions, we are actually bang on time. And I must share that it's just been 2 years since I met Dr. Rigoutsos in Dr. Derman's office. And it was a half-an-hour meeting. And I mean, I will always remember it in my lifetime, the kind of joy that Dr. Rigoutsos had and the way he's been guiding our team. Thank you so much, Dr. Rigoutsos. Your guidance and mentorship is invaluable to our team. And I would, on behalf of all of us here at IHOPE and LVPI and Jefferson, we'd like to thank you for taking your valuable time to join us this evening to share your amazing work and knowledge with us.
Well, thank you for your kind words. Believe me, the pleasure is all mine. Ever since that meeting in Richard's office, I think I told you at the time, I said, it's very infrequent that I say something to a colleague and they say, oh, yeah, of course. And that's what happened with us. And I'm like, perfect. So, you know, and here we are. And we have results to show for it. So it's great.
Yes, shortly, yes.
Thank you so much, Professor.
Dr. Raja, please would like to.
Thank you so much. I echo Vipin's sentiments. And thank you for sparing a valuable time, not just for this webinar, but for all the collaboration that you're doing with LV Prasad Institute. I'm sure, and I'm hopeful also that we will have many more such collaborations in the future with you and Jefferson. Thank you so much. Thank you.
Thank you for the invitation.
And my office is always open.
Thank you, Dr.
It is.
Thank you.
Absolutely.
Thank you so much.
Thanks again, everyone, and have a good weekend.
Thank you.
All right. Good night. Thank you, everyone. Bye.
Bye.